You're listening to the Common Descent Podcast.
Hello, David.
Hello, Will.
And hello, listeners. Welcome to episode 223 of the Common Descent Podcast. Today's episode, we are discussing mutations.
Mutation. Said in the voice of Patrick Stewart.
Yes. All of you need to hear it that way. I'm very excited about this topic. This is one of those foundational topics. This is key to understanding how life works on our planet and how evolution happens.
Yeah, we get to get into the genetic weeds a little bit.
Yes, so get ready to jump in. This is going to very much be an overview topic because this is a large topic, but it will be kind of this one feels the most to me like we are effectively recording an intro textbook chapter. So we'll go over what a mutation is, what causes mutations, what kinds of mutations those things can cause, how we are protected from mutations, and then how it applies to evolution and to paleo research. How do we study mutations in the past and use mutations to understand the past? This topic was requested, which is why we're talking about it, as all our episodes are. This was requested by Bobby, Maeve, Ryan, Jackie, Hannah, and Eli.
Thank you, everybody, for your request. Hey, listeners, if you have a request, there's a topic requests form on our website. Link in the episode description.
Absolutely. Before we get into our main discussion and before we get to the news, some quick announcements. First and foremost, as always, we have a Patreon. The donations from patrons on our Patreon fund the podcast top to bottom. It allows us to do all the cool things we get to do. And if you sign up on Patreon with us, you can get extra goodies, bonus episodes, bonus content, live streams with us, etc., etc. So you can find that link down in the description. And if you sign up at certain levels, you can get your name shouted out here on the podcast. So today we would like to welcome Amy, Beth, Whitney, Caden, Nekonomicon, Ollie, Robert, and Matthew.
Thank you all so much for joining us. We appreciate your support as always. This summer, we extra a little bit have appreciated the support of many of our patrons because this episode is released right after Snake Month is over. We are recording this right at the tail end of Snake Month.
Yes.
Which means by the time this comes out, the Snakes and Crocs tier will be closed. But we got a ton of participation on our Snakes and Crocs tier on Patreon this summer, and that support contributes to charitable donations that we will be making to further reptile and other wildlife conservation. This summer, in total, more than 100 people participated in helping us to build up the funds for making these donations. So we can proudly announce that we will be donating, in honor of Croc and Snake Month, $500 each to the Crocodile Research Coalition, Save the Snakes, and the National Parks Conservation Association.
Yes. It feels just so wonderful to have another successful Croc and Snake Month. And thank you to everyone who participated, who helped us make these donations and showed their support for these critters.
Yes, huge thanks to everybody who contributed financially. And even if you are not one of the people who was able to make those financial contributions, thank you so much for participating and engaging. If you joined the live stream, if you joined the Discord Q&As, even if you just listened to and enjoyed and maybe shared around our Croc and Snake themed episodes, all of that helps make Croc and Snake Month an event every year, which is what allows us to give back a little bit to conservation efforts, things like that.
Yes, so thank you, everybody, and look forward to next year to another Croc and Snake Month.
Speaking of cool podcast events and participation from our audience, very recently, Nora and I did a museum meetup at the Royal Tyrrell Museum.
Yes.
which was extremely cool. Huge thanks to everybody who showed up. There were a ton of people. It was awesome. Huge thanks to everyone who joined and said hi, and an extra big thanks to the people who joined us who worked at the museum, who helped. I was one in particular I want to call out, Ben, who met up with us, stuck with the group as we explored the whole museum, was in tour guide mode, even gave us some inside info, showed us a couple educational specimen places, things like that. Ben was awesome. Also, Ben helped to jump our car the next day when it wouldn't start. So listen, I'm not saying I have favorite listeners, but great work, Ben.
It's pulling double duty.
It was awesome. We will be doing more museum meetups. We're planning to do one in Atlanta in October. More details to come later on.
If you want to see, there's a bunch of pictures that people shared from the meetup in our Discord. So if anyone's interested or didn't get to see that those were shared, you can find them in our Discord.
Absolutely. Also, people who came to see us at the museum meetup weren't just fans of the Common Descent podcast, but also of Leave It to Us, of which Nora is the main host, along with our friend Dr. Ali Baumgartner. Leave It to Us is in the second half of its first season as of the release of this episode, and at the end of season 1, they're going to be doing a mailbag episode. Patrons on the Leave It to Us Patreon have the opportunity to submit questions for that end of the season mailbag episode. So if you would like to do that, go do that. Also, we got mail.
Yes, we did. We got some cool mail. We got sent a book by Terry Pratchett, The Last Continent, from Caitlin, who noted that we had not mentioned Sir Terry Pratchett in any of our nerd discussions, and they thought that was an oversight, so they wanted to send us one of their books.
Terry Pratchett has a gray fossil site plant species named after him.
Oh, right. I forgot about that.
Cavalignum pratchettii.
Yes.
Thanks to Dr. Liz Hermsen. Also, we received, and this was not mailed to us, but handed to me in person at the Royal Tyrrell Museum, a couple of little watercolor images that each one has a sketch of a fossil croc or snake, and then a watercolor image of the environment that they lived in from Isabella, who was one of the many delightful people that showed up to the museum meetup.
They're real pretty. Like, they're really cool looking. So thank you to both of you for those things. Caitlin also assured me that there are audiobooks of this book for me, who does not like to read. So I appreciate that.
Excellent. Hey, one more thing to announce before we get into the actual meat of the episode. This episode is being released at the beginning of August, and at the end of August, we're going back to DragonCon.
DragonCon!
DragonCon takes place in Atlanta. This year, Will and I are going to be on almost a dozen different panels and events and things. The final schedule isn't quite finished yet, but once it is, we'll post it on our social media and we'll share it on Discord and stuff. But we can tell you for now that we will be participating in things that include, but are not limited to, the hands-on science activity space, panels about science after dark, de-extinction, that's going to be a fun one. We will be enjoying, once again, science versus movies, where also I'll be participating in the first ever nerd night hosted at DragonCon, which is a very exciting. There's all sorts of cool stuff. We are going to be having at least one panel, I think, maybe every day of the con this year, starting Thursday, August 28th through Monday, September 1st. If you are in the area, come check it out. Say hi to us. DragonCon is tons of fun.
It's going to be so much fun, everyone. If you're there, come say hi. We are looking forward to it so much.
Also, DragonCon weekend has historically been roughly when we announce the upcoming spooky theme.
Yes.
So that's another thing to look forward to around the end of the month.
Soon. But for now, we will get this episode started with the
jump right in.
section. Let's jump right in, as the podcast tradition goes. Every episode, we like to gather up some recent scientific studies in the fields of paleontology and evolution and earth science and share them all with you so that we can all stay up to date. David, what's your news?
My first news is about Neanderthal tools.
Cool.
Specifically, tools made from bones of some pretty cool animals. This is research by Gregory Abrams et al. In the journal Scientific Reports, and in the blog post, we will link to an article in Popular Science by Andrew Paul. Neanderthals are our famous extinct human cousins, episode 177 for all sorts of stuff about that. There's a lot that we know about Neanderthal lifestyle and history and hints of their culture. There's also a lot that we don't know. One of the things that we don't have a ton of information about is how Neanderthals interacted with large predators in their habitats.
Mm.
This paper spends a bunch of time talking about Neanderthals and big cats because there were a bunch of large cat species that they overlapped with in their time and space. There are some examples that have been published previously of the remains of lions, cave lions, and even saber-toothed cats that have signs of butchery on the bones where they might have been carved up to be eaten by Neanderthals, or bones that seem to have been carved into some sort of tool, which is something we see a lot in ancient human sites where you have bone tools. This new paper describes the oldest example of a bone tool made from the bones of cave lions and some really interesting insights into how they were using these tools.
Cool.
These are remains from a cave called Skladina Cave in Belgium, which is a previously known cave occupied by Neanderthals. There's a bunch of Neanderthal stuff in it, including a whole bunch of animal bones that have been crafted into various tools. This includes 4 fragments of cave lion bone, which sounds like are mostly limb bones,
Okay.
that are dated here to as old as 130,000 years. The age is exciting. The fact that they were made from cave lions is cool. But one of the things that's particularly interesting about this is that the bone tools show evidence of having been multifunctional.
Oh, okay.
The paper describes in detail, for example, a tibia that has damage. I think they described one end of it as sort of beveled, and the shape of it and the wear on it suggests that it was used as a chisel or something of a similar chiseling function. But then later, the bone seems to have been broken and recarved into a flint crafting tool, a
Huh.
retoucher tool. And they also describe one surface of the bone that has a different type of wear that seems to be from some other thing that it was being used for that isn't quite clear what it is, which means that this was a multi-tool.
Cool.
That this now, how many of these? It sounds like in some instances this might have been it was this one tool and then later was reshaped into a different tool, or possibly it had multiple functions at once. But this is a really fascinating look into Neanderthal technology and creativity, right? This wasn't, again, dispelling the old notions. This wasn't just that they picked up a sharp bone and used it to do stuff. They were crafting with this intentionality. And in the paper, they also mentioned they did a statistical analysis on the shape of the tools. Specifically, they were talking about the retouchers, which are used for crafting flint. And they found highly consistent dimensions, the shape and size of these tools, even though they were made from a variety of different bones, ribs, leg bones, other things, from a variety of different animals.
Yeah.
So even though they were getting them from different sources and different starting points, they were consistently making them the same type of tool, the same shape and proportions, which suggests a consistency and uniformity to their tool making techniques.
Yeah. That's very cool. I like this because A emphasizes that yes, they were advanced humans, like they were doing human stuff.
Mm-hmm.
The consistency of the tool shape is satisfying because that both makes sense from if the tool's doing the same job, then even if different people are coming up with it, they'll probably come to similar shapes. But if it is that consistent, they're probably learning from someone else who made the tool.
Yeah, and they've refined their technique.
Yes.
That this is they had a right way to do it.
Yeah. The multi-tool function of it makes me happy because even if it wasn't like this tool was designed to do all both of or all three of these jobs. It's like when you're like anyone who's been doing housework and maintenance stuff and you're screwing something in and then you need to just tap it in, you should go get the hammer because that's the tool for that job. But the butt of the screwdriver will act as a temporary hammer for a very
That'll
quick
work.
job. And it's like that's using something as a multi-tool, but here we see that there actually seems to be some features leaning toward that, which is very cool.
One last little note, the authors do discuss a bit in their study about the question of how the Neanderthals got hold of cave lion bones.
Mhm,
And
mhm.
unfortunately, there is not a clear, obvious evidence to the answer. It's entirely possible that they found a carcass and scavenged it
Yep.
and then used the bones. It's possible that they found bones from an already decomposed carcass, although I think there was mention in the paper of the damage on the bones looking like it was fairly recent after death,
Yeah, yeah,
that the
yeah.
bones might have still been fresh. And it is entirely possible that these Neanderthals hunted and took down a cave lion and then probably ate the meat off of it and used the bones. So how, what story led to them having a handful of cave lion bones? We don't know, but there's a bunch of different possibilities.
That's so neat.
Neanderthals are pretty cool guys.
Well, my news also has to do with predator interactions because it is about a bite mark on a terror bird bone that seems to be from a caiman in South America.
Ooh, this is one of those Jurassic Fight Club
Yeah,
scenarios.
yeah, this definitely has that vibe to it. This is research by Andres Link et al. In Biology Letters, and the article is a press release in phys.org by Crystal Castle. This is an exciting find because direct evidence of interactions between animals, as we often like to note, are rare. When we have trace fossils, it is exciting when we get bite marks and so forth because that is direct evidence of two animals, two organisms interacting. But they note that this is an especially rare example of that where it is two top predators,
Yeah.
and that is something that does not preserve evidence very often and is just a less common phenomena. Predators do not tend to interact as often as they are interacting with their prey, so getting information as to what kind of interactions go on between big predators is interesting and important. This is from the La Venta fossil site in Colombia, which is a site that preserves fossils from the Middle Miocene. So this would have been the Pebas super wetlands system that used to exist in South America. We talked about this in the alligators episode because that's where caimans went crazy during that time.
Yes, episode 194.
They found the left tibiotarsus, which is a leg bone, of a forest wrassid, which are terror birds. And this was from a large terror bird, it sounded like. I don't know if it was like one of the bigger ones, but not a small member, what would be a decent sized predator.
Sure. And terror birds, as a reminder for our audience, were large, flightless, predatory birds,
Yes.
mainly known in South America, discussed in episode 37.5, which is a thing
Yes,
we
yes.
only did that once.
Man, that's one of the first times that one's gotten to be called out.
Uh-huh.
They found 4 pits on the outer bone of this leg bone and noted them as tooth marks. There's no sign of healing, so this was not a survived attack. This was something that happened either while the animal was dying or shortly after it had died.
Hmm.
3D surface scanning and digital modeling of these marks allowed them to match it up to likely biters. And they found that the most likely candidate for the bite was a middle sized caiman. And the a potentially good candidate for that would be Protosaurus navensis, which is not the big species.
Right.
Protosaurus bristialis or something. Brasilensis is the big, potentially 30 to 40 foot species of Protosaurus. This is a much more moderate size. I don't remember what size it gets up to, but is a more normal crocodilian sized caiman. They don't know for sure that this was Purosaurus, but that is a correctly sized member that is also known from the La Venta fossil site. So was definitely sharing territory with this species of Forest wracid. Whether this is a predation or scavenging event, once again, that is very hard to know. Both are very possible. It could be that this terror bird had come to the water either to hunt other animals coming to the water or to drink itself, which would put it in ambushing range for a caiman. But caimans are well known scavengers, so we won't know whether the bird was alive when it was bitten or if this was just a scavenging event. But if it is indeed this species of Purosaurus, it does contribute to our understanding of what its diet entailed,
Mm-hmm.
which is fun, and indicates potentially if this was a hunting event, it informs the relationship of where large terror birds in this wetlands actually might have fallen, that they were on maybe have been at higher risk of predation than previously might have been suspected for a large predatory animal.
Sure. Well, and it's, a terror bird would not be hunting a medium-sized caiman.
Nope.
That doesn't seem like the kind of prey a terror bird would be good at going after. So this seems like a very one-directional predatory relationship.
Though, if I were a betting person, I absolutely think baby caimans would be on the terror birds menu
Oh, for
because
sure.
hunting birds, especially like striding birds, go after them all the time.
Yep.
So this may be one of those where it's yeah, when I'm little, I'm on your menu, but once I'm big, you're on my menu.
Yes. I'm always fascinated whenever we find crocodilian tooth marks on a fossil animal
Hmm.
because my general assumption is that if a bone got swallowed by a croc, we won't find that bone intact as a fossil,
Yes.
which would seem to suggest that the leg bone got bit but not eaten
Yes.
by the croc, which raises all sorts of questions. Like, was this something that you bit it and then realized you didn't actually want to eat that and let it go? Did you bite and then drag it into the water and then let go and get a better hold and swallow, most of the rest of the body? Did you bite it and then someone else bit the other half of it and you twisted it and like tore it apart? What was the circumstance that spared this bone after the initial bite?
Yes, exactly. The third scenario was immediately what came to mind was if you grabbed what you pulled off a leg, part of a leg, and then you went away because the body was being occupied by a bigger caiman or who actually killed the terror bird and was
Mm-hmm.
eating it, whatever it was, and those bite marks are where you came and pulled a piece off, but that
Right,
piece remained, and then whoever finished eating it didn't eat bones. So
right.
the bones got left.
It could also be that it was a bite that was an unsuccessful hunt, but a successful killing bite.
Yes,
Right.
exactly.
Maybe you tried to take down this bird, got a hold of the leg, but then it got away for whatever reason. But then the bird died shortly after that. There's
Yep.
all sorts of different ways that this interaction could have played out, which is very fun
Yes.
to think about.
Yeah.
Well, speaking of trace fossils left behind by predatory reptiles, I've got news that is about dinosaur dancing footprints.
Nice.
This is research in Cretaceous Research, the journal by Rogers Bunton et al. And we will link in the blog post to an article in Live Science by Patrick Pester. This is not the first time that we have mentioned this phenomenon on the podcast. In addition to dinosaurs leaving behind footprints from walking and running, there are also less typical behaviors. There are footprints of dinosaurs in shallow ocean sediments that are interpreted as swimming traces. There was one paper that came out a little while ago. I think we talked about it on one of our things of a footprint trackway of a small dinosaur that seems to be a takeoff
Yes.
track of it running and jumping a couple times and then taking off into the air. There are a handful of examples of unusual tracks that have been attributed to potential courtship displays. In modern birds, there is a relatively common behavior called lekking. And in a lek, typically this is male birds putting on display performances for females, where there's often a particular little like arena. Sometimes the birds clear out the space intentionally for this, where the male bird does a bunch of fancy footwork and flapping the wings and doing all sorts of things. Birds of paradise are very famous for doing this kind of thing, but there's also a bunch of other, especially ground dwelling birds, that will have a little dance floor for showing off to mates.
Yes.
There have been a number of fossil sites that have unusual footprints that have been interpreted as potentially non-bird dinosaur lekking sites. These traces include an ichnogenus, a type of trace fossil called Ostendicnus, which are, generally speaking, scrape marks. So it's not like a footprint. So I think the common form of it described in the paper is it's a pair of grooves sort of into the sediment with a ridge between them that is toes dragging across the sand. Sometimes they're associated with claw marks as well. So it seems like a poking the toes into the floor and then dragging them along the ground. This could be intentionally moving sand around, like digging out a nesting site. I believe there are some birds today that just kick up sand as part of their display moving around. It could be active nest building. So there's a bunch of different reasons why this particular trace might be made, all of which fit with what we see in birds today when they are courting or preparing a nesting site. Examples of these traces are known from multiple sites in the late Cretaceous of Colorado and at least one site in the early Cretaceous of Alberta. This study describes new records of these scrape marks at Dinosaur Ridge in Colorado, about 100 million years old. They actually mapped out the tracks using drone imagery. So they flew drones across over the rock surface and took scans of the footprints. They described traces from two different rock surfaces, one of which had a single scrape track on it, and the other one had 24.
Wow.
Based on the size and spacing of these tracks, the authors interpret that this was roughly an ostrich-sized theropod, which is a two-legged dinosaur. Ostrich-sized is pretty moderate-sized for most Cretaceous theropod dinosaurs, so this could be like an ornithomimus-sized or oviraptor-sized, somewhere in that. It could also be like a small tyrannosaurid. There's not any specification on what it might be. There also seems to be potential evidence of multiple individuals using this site. In the article, the authors are quoted as describing the patterns in these tracks and the movements that they suggest, that in some places it looks like the dinosaur was stepping side to side as part of these scraping steps. In other cases, they seem to have been scraping while backing up. There are some cases where some scrapes are interrupting earlier scrape marks, where they're sort of moving over the original scrapes, all of which seems to show this like back and forth, somewhat patterned movement that might be consistent with putting on some sort of courtship display.
Very cool.
They also note that because there are scrapes on multiple stratigraphic levels, multiple layers within this site, that suggests this was an area that was being returned to over time for use as this site. Overall, the fact that there are now, this is, I think, the fourth location that this type of trace has been documented, seems to suggest that this was not an uncommon behavior. That if this is in fact some sort of courtship nest preparation behavior like we see in modern birds, it seems to have been present at a variety of places in a variety of times. this just adds, this is what I've been saying for years. Dinosaurs were putting on goofy dances. They must have been.
Yep. Yeah, no, that's exactly what that seems to entail, which is fantastic. Like, that's so satisfying. That's so lovely to think about. And what the markings with what'd you say, 24 marks?
24. Yep.
That's awesome. That is so cool. Well, and it makes you wonder of like, yeah, was this an area where you were competing with each other? So there were multiple individuals all vying for attention and vying for the dance floor. And
Mm-hmm.
so, you were fighting over this spot, or was this being used multiple times by different individuals because this was the best spot? This had the best point of,
Yeah.
viewpoint, or got the best lighting.
There's a ton that we don't know. So we don't know necessarily how many were using it. We don't know if like did courtship happen here and then mating also happened here,
Mm-hmm.
or
Mm-hmm.
were you nesting somewhere nearby? Also, it is very reasonable to assume that these were males doing courtship dances because that's what we see most of the time in modern birds. But that doesn't have to be the case. It could be
Yes.
that these were females doing the dances or that both sexes were doing the dances. So there's a lot of open questions. And there was actually, I think this was somewhere in the paper that was talking about a lack of copulation trace fossils to tell us like where mating was happening, which does get me thinking about what would copulating dinosaur footprints look like.
Yes.
And I guess it would look like the two dinosaur footprints that get real close to each other.
Yep. Mm-hmm. Mm-hmm. That did not leave room. Did not leave room for Jesus.
This dinosaur peculiarly seems to have had 2 left feet and 2 right feet.
Yes. Yes.
I'm not enough of a biblical person that there's some sort of joke about there not being enough room for one set of footprints in the sand.
Oh yeah. Yep. Yep.
You can enjoy concocting what that joke should be.
That's so neat. Well, my next news also is about ostentatious displays.
Oh, fun.
This is a study about a ancient reptile that has feather-ish display structures on its back that aren't feathers but are surprisingly feather-like. This is research by Stefan Speakman et al. In Nature, and the article is another press release from phys.org. So this study is looking at a ancient reptile from the Middle Triassic that has a row of appendages coming off the back that are these very display looking and kind of feather looking structures, which is notable because complex skin structures, integumentary appendages, as they call it in the paper, are only really known from birdline with feathers, so dinosaurs, and mammals with hair.
Right.
That kind of complicated and out of the skin structure is not known in other groups. And this seems to be that kind of structure, but not feather or hair, which is exciting. Specifically, this is Mirasaura grauvogeli, which altogether means Grauvogel's wonder reptile. This is a small animal. So once again, from the Middle Triassic, this one dates to about 247 million years old. It is related to a group known as the Drepanosauromorphs, which is a famous group, the Drepanosaurs, that are these really weird group of reptiles that are kind of generally lizard-shaped, but have a very bird-like face, like very pinched, sharp snout, these powerful gripping forelimbs with often big thumb claws that are almost like velociraptor claws, a seemingly prehensile tail, sometimes with a claw-like horn at the tip for grabbing onto branches, it seems, and these very chunky barrel-shaped bodies, these very stout bodies.
Yeah, Drepanosaurs are most famous for being confusing.
They're very weird. This is a member of that, and this one has a row of not spines, but these display appendages coming down the middle of the back. They compared it to Longisquama, which is one of the famous ancient reptiles that has a back sail of separate pieces.
Yes, when you started talking about this one, that's what I assumed that this was about. That
Yep.
this was about Longisquan. I couldn't remember the name of it, but that was what I figured this would be talking about.
Yeah, when I was reading the paper, they were like, "It's very similar to Longisquama." I go, "I don't know if that's the name, but I assume you mean that other one I know."
Yo,
This is a new species that has a similar display structure, but these are very feather-like. They have a ridge with this kind of feathery like structure coming off of the ridge part, and they differ significantly from the scales that we would expect to see on a reptile like this. Taking a closer look, they do note that though they are like superficially similar to feathers in many ways, structurally they are not feather like. They don't have any of the branching that feathers are known for. So they don't seem to have a similar structure or development as feathers. So likely these evolved independently from feathers. This specimen does preserve melanosomes, which are the pigment containing organelles of colored cells, that gives body skin its color. Looking at these and looking at the morphology of these melanosomes, you can compare them to similar melanosomes from similar structures. And they found that these do have similar geometry to melanosomes from feathers
hmm.
and do not represent melanosomes from skin or from reptile skin or mammal hair. So they do seem to have more similar coloration structures to feathers, but once again, the lack of structural similarity in the actual appendage and the placement of this group, the fact that it is nowhere near being related to birds, suggests that this evolved separately from feathers and hair and is another unique type of integument structure that we hadn't known about previously, which is very cool.
Fascinating. We talked about episode 183. We went through feathers and what we know about their evolution and what makes them distinct. It has been said by us and by other scientists for a long time that it would not be surprising to find, especially in like the Triassic period, a variety of feather or fur like structures on ancient reptiles. And as that looks more and more like a possibility, it starts to raise questions about our assumptions about how good representations our modern reptiles are
Mm-hmm.
of reptiles over time. Because it's very easy to say, well, modern reptiles, discounting birds, don't have any feathery or furry, like they have bristles sometimes in some lizards, but that's not real, like spiny things. But they tend to not have complex integument, which is true. But then you can also stop and think, well, crocodilians are aquatic
Mm-hmm.
and turtles have shells, and lizards and snakes are typically and ancestrally ground dwellers
Mm-hmm.
and often burrowers. So it's very possible that we just happen to have living today groups of reptiles that evolved out of the potential for this kind of integument. But if we
Yes.
rewind the clock 200 million years, that might be more unusual than we would expect.
Yeah, exactly. They noted that, like you were mentioning, the idea of complex skin structures having a earlier origin, and having ties back to earlier lineages. They noted that developmental studies have suggested that likely the at least genetic basis, like the genetic ability to start growing these kinds of structures, probably dates back to as early as the Carboniferous, 300 million years ago. But this is our first early evidence of this kind of structure. Not going back that far, but still, this is our earliest evidence that, yeah, there were early animals with complex structures coming off the skin other than feathers or hair. And so, not only does this suggest that those kinds of structures are not restricted to the bird line and mammal line lineages, but also that there may have been other kinds of structures that we just haven't yet found evidence for or recognized as evidence for.
Very cool. The Triassic period was a bizarre place.
Oh yeah. I love the idea of a whole bunch of display structures that are made out of things that are would be new to us.
Yes, the idea that in the Triassic there were no birds, but there were potentially a bunch of tree slash ground dwelling reptiles doing similar displays
Yeah.
to what we see in birds today.
And on the note of interesting and unique genetic features, this leads us nicely into that's a segue I could have made for just about any news
Any
we ever
news that
did.
we've ever talked about. Speaking of evolution,
let's talk about mutations and what that actually is and what kind of effects they have. Mutation is another one of those topics that we are getting to late into the podcast, but is actually a very foundational concept.
yeah, this is like literal textbook subjects in biological sciences.
Yes, mutation is a common topic just discussing genetics, DNA
Mhm.
and your genome and the processes that go along with that. Mutation is a key part of understanding that. It's also foundational to how we understand evolution and how evolution functions and is driven. Mutations are a key aspect of that.
Yes, you might say, here's what I know about mutation. It is the key to our evolution. It has allowed us to evolve from a single celled organism to the dominant life species on the planet.
And we'll get into that later in the discussion. But first, let's do some definitions. Just make sure we actually have all of our terms. A mutation is actually a very simple concept. It is a change in the nucleotides of a sequence of DNA.
Yeah, the A's, C's, T's, and G's.
Exactly. Nucleotides are those pieces of code, guanine, adenine, cytosine, thymine, that pair up with G's and C's and A and T's to make the code of our DNA. And the order that those are in get read in usually short bunches, little triplets, codons that code for proteins, and those then make the different functions that our cells do. A change in one of these letter pairings is what a mutation is. Now, it could be a single one, it could be many. We'll get into the diversity of mutations.
Sure. But basically, a mutation is a DNA typo.
Exactly. It is something has gone wrong or awry, and the DNA code is not what it used to be. It is not what it was before the mutation happened. And if that sets in, that can now become a permanent change. You'll sometimes hear the term allele come up in regards to mutations because alleles are alternate forms of a gene to different or maybe multiple different versions of a gene. This is where we get like the dominant and recessive versions of genes and things like that. These would have at some point been the result of a mutation that caused you to have these multiple versions of a gene.
Right. That this is an interesting part of this conversation because the whole idea of a mutation is something is off, something is atypical about the sequence, the genetic sequence, and alleles are a case where you have multiple coexisting and often functional versions of a gene that are just somewhat different. That's not technically a mutation because it has become a regular and standard part of the genetic code of that organism.
What at some point was a mutation and is now fixed in our DNA. It is now just a part of the genome, but at one point it was a mistake that happened
Yes.
and caused this new allele to show up.
Which, funnily enough, arguably is true of our entire genetic sequence.
Exactly. And we will be touching on that as we go through the discussion that mutations are the one of the main ways diversity is introduced into genetics. There are others, recombination and transferring of genes across genomes, things like parallel gene transfer in bacteria. Like there are other ways to get diversity and rearrangement of genes, but mutations are one of the key tools that is utilized by evolution to have diversity and new traits to work with. But first, let's talk about what causes mutations and what kinds of mutations there are. Because there are different ways things can mutate and different things that cause these different types of mutations. Two most common groupings of mutations is point mutations and chromosomal aberrations. Point mutations are exactly what they sound like. A single point is mutated. One letter pairing. One of
Yes.
those pairs of nucleotides is messed up somehow, and the single point in that DNA sequence is altered.
Right. Where there was meant to be a C, there is now a G.
Yes. These are extremely common form of mutation and typically show up during DNA replication. So while we are making a new strand of DNA, you have to copy all of the base pairs that are go to either strand of the DNA as you make your new strands. And during this process, the wrong letter could get put down and you have a point mutation where that wrong letter got put down.
Right. And it's important to remember that DNA, we're talking about individual nucleotides, the letters that make up a DNA sequence. There are billions of those in our
Yep.
DNA. There are so many of them. And DNA being replicated isn't just something that happens when you're having babies or something. Every time a cell divides, DNA is being
Exactly.
replicated. And that is a thing that happens in your body all the time. So the number of times that a single nucleotide pair is replicated over the course of your lifetime of a body like a multicellular animal's body like ours is unfathomably large. There are so many. Like if only a tiny, tiny, tiny percentage of the time an error happens, that's still going to be thousands to millions of errors.
Yes, exactly. It's a genetics telephone game. Every single time your cell has to divide, it needs to accurately copy the entirety of its genetic information. So every single time is a chance for a mistake to happen, and those mistakes could eventually slowly build up. And you could end up with some really crazy mutation situations going on that lead to effects that we'll talk about later. Chromosomal aberrations are more drastic, large scale mutations. This is usually when something happens and parts, entire sections of chromosomes are affected or altered. This will happen during things like meiosis, which is the splitting of a cell into reproductive cells, germ cells. And when you split the chromosomes into their two halves, sometimes a chromosome might take too much from the other side and it doesn't split perfectly in half. You can also get during recombination stuff slippage where the code shifts along the line and is matching up differently than it was before. Like
Yes.
the letters haven't changed, but the way they're meeting up has.
It's like missing a button on your shirt, and
Yes.
and you've just your whole shirt is just lopsided now.
Yep. You also see different categories of mutations specified in multicellular organisms like us between somatic cells and germ cells. Somatic
Yes.
cells are your body cells, muscles, skin, nerve, all of the just parts of your body. Those are going to stay with you. When you have a baby, none of those cells are going to be parts of the cells that go into making that baby.
Right.
Those are germ cells. Germ cells are the reproductive cells. So mutations in a somatic cell, a somatic mutation only affects the individual it happens in. A mutation in a germ cell is the one that can be passed on to the next generation. So only
Yes.
mutations in those specific cells or cells that will become those cells are the ones that actually can be passed on. So only a very specific percentage can actually be transmitted.
In our case, those germ cells are sperm and egg cells. But if you're talking about plants, if you're talking about whatever organism you're talking about, whatever cell it is that is going to become a seed or an embryo or whatnot, that is the genetic material that is going to be passed on to an offspring. And in many cases, that one cell is going to give rise to the billions of cells that will make up that new organism's body.
Yes. And so if a mutation happens in a germ cell, every cell in that new individual that is born from that germ cell will have that mutation. And so that's why it can be have a more drastic effect than it seems like if it's happening in such a small percentage of cells. But every time it does happen, that entire new human or animal has all those mutations. The way mutations happen, what causes them, the two most common ways and the two typical ways you will see is mismatches during replication and
Mhm.
mutagens, which are outside effects, chemical or physical, that cause mutations. Mismatches are errors during replication. So,
Mhm.
this is by far the most common form of mutation that we come across because we are constantly, as we just said, replicating cells in our body all the time. As you grow, as a new organism is growing, their cells are multiplying exponentially. During this replication process is when most often errors occur and a mutation happens. There are a bunch of different ways this can go about. Some are caused by the replicating agent itself. So, polymerase is the enzyme, DNA polymerase, the enzyme that attaches to a DNA strand after it's been unwound by helicase and other enzyme partners and protein partners. It attaches to a strand, and it's the one that moves down the strand and grabs nucleotides floating around in the cytoplasm of your cell and attaches them to one half and slowly makes a double stranded piece of DNA into two double stranded DNAs. While it is placing down nucleotides, it might place the wrong one down. Placing just an incorrect one down is the most common mistake. That's mismatch, point mutation. These are most common to happen between the two types of nucleotides we have. A and G are purine nucleotides, and C and T are pyrimidine nucleotides. But usually, purines will be switched for purines, or pyrimidines will be switched for pyrimidines. So you'll get a switch like that where the nucleotide looks kind of similar and got grabbed mistakenly.
Right. Because it's important to remember that all of this is functioning based on the shape of molecules.
Yes,
Right. That's the way that that enzyme functions is it interacts with molecules in the cell. And based on the shape of that molecule, the interaction varies. And if it's the right shape coming in from the outside and it's the right shape on the other side, the enzyme can put those two together. And
yes,
A and G are different. Adenine and guanine are different molecules, but they are a very similar structure. And so you
yes.
can get one being grabbed when the other one was more ideal or more intended, as it were, nucleotide to be put down.
Exactly. This is actually a very common mistake that happens. I saw the main source I found most of my notes from noted that as far as just a chemical reaction goes, the polymerase selecting of nucleotides and base pairing nucleotides is actually not very accurate. Like, if you could just run it in a test tube, which isn't something we can actually do, but if it could just be run purely chemical reaction wise, it would probably have an error rate of about 5 to 10%,
Okay.
which isn't like 50%, but that's
That's a
notable.
lot. Yeah.
Yeah, if 5 or 10% of my paycheck is missing, I'd notice. That is a notable amount.
If 5 to 10% of my DNA is different, I'm a fish.
Yes, yeah, yeah. So like these mistakes happen quite regularly. They also don't happen evenly on the two new strands of DNA.
Mm.
So when polymerase is copying down onto these two halves of the DNA, one strand, so DNA has a front and a back end basically. A 5 prime and a 3 prime is what they get noted as, but they have a front and a back. And polymerase moves one direction along those. So as helicase is moving down it and unspooling the DNA, one polymerase can just follow behind the helicase and copy continuously, but the other strand has to backtrack. It has to helicase has to unwind a little section, and then polymerase copies that section, and it unwinds a little section, and then it copies that section. And they call it a lagging end because it lags
Mhm.
behind. The lagging end tends to have notably more errors, upwards of 20 times as many errors. Now, whether or not this is due to the lagging feature of the replication, or if that polymerase, which is polymerase 1, is less accurate than the polymerase 3 that does the main replication. Either way, your two new strands are not built equally. One is more likely to have more errors than the other.
Yeah. Which is a very interesting. We're already starting to lay the seeds of a topic that I'm sure will continue to come up throughout this discussion, which is that mutations are not fully random.
No,
There
they are not.
are patterns in where certain types of mutations happen, when and where we might expect more mutation than at other times. This is one of the biggest, right, the misconceptions about mutation is that it is a totally random, unforeseen, unpredictable thing that happens, and that's not quite true.
There are actually notable patterns in it. Another way that base pairs can be mismatched, and I like the way they said this, where polymerase is not to blame, is that sometimes the base pairs can shift. So your base pairs have different phases that they can be in called tautomers,
Mm.
and these are just structural alternatives that it's still the same base, but its molecules have shifted a little bit and it has a different state it's in. These typically have one that they are in most of the time, but then another they switch to every now and then. Doesn't affect anything, doesn't affect how your DNA functions typically, but if that switch happens while they are about to be replicated, they get read differently. For instance, thymine has a keto and an enol tautomer form. The keto is the normal one and it pairs with C, but the enol form pairs with A.
Oh, interesting.
So when it gets read, polymerase goes, oh, this wants an A nucleotide because of the enol form it's in. Adenine also has two forms, and in its amino form, it pairs with C instead of T. So if the nucleotides shift at the precise moment they're about to be replicated, polymerase puts down the one it's being told to put down, and then after replication goes on, the base will revert back to its normal form, and now it is mismatched. Now it goes,
Yes.
what is this T doing here? Where's my C? And now you have a mismatch. Now there is a mistake. You can also get mutations during replication that aren't a mismatch, but a deletion or an insertion.
Yep.
Something is not put down, or an extra is put down. And those can cause their own kinds of mutations.
Yep. You skipped a C or you put in an extra C.
Exactly. These are often called frame shift mutations because they inevitably bump the line down. It's like it with your shirt analogy, but instead of just not buttoning, one side now has too many buttons.
You put an extra button on, and now all the
Yes.
buttons are wrong.
And now you can't line it up correctly. So if this happens within a gene, something that codes for a protein, and you add something in there, it's going to shift things off for that gene and the genes around it.
Yes. It's like when you were in school and the teacher would be like, all right, get in two lines and I'll pair you up. And you can see where your friend is on the other line. And you put
Yes.
yourself you put yourself into the line to line it up perfectly. And then the teacher moves somebody.
Yep.
And I was like, all right, well, why don't you move to the end of the line? It's like, well, now the whole line has shifted and I don't get to be with my friend.
Exactly. Now, if it doesn't happen within a gene, then it's not going to have the frame shift effect. So not all of them are going to be this frame shift mutation. And if it inserts or deletes three nucleotides or a multiple of three, then it's doing whole codons. And
Yes.
that will be not quite a frame shift because it will just line things up with another set of codons.
Yes. So for people who, if you don't remember the genetics basics, three base pairs could come together to code for an amino acid.
Yep.
And depending on what that order of bases is, changes your amino acids, and then amino acids are the molecules that come together to make proteins. So
Yes.
if you right, if your codon of three base pairs, if one of them gets misplaced, your amino acid is going to be different. You're going to get a
Yep.
different structure. But if all three of them get misplaced, you just don't get that amino acid. You just skipped
Yep.
that whole chunk.
So if it's in those multiples of 3, it is still a mutation, and it will still change things, but in a kind of a less messy way.
Yes.
These are most common insertion and deletion mutations in sequences of repeated code. When the code is
Mm,
repetitive, these are sometimes called microsatellite sections of your DNA, where there's just very repetitive sequences of the genome. Here, it's really easy for basically the reading to slip and to me it feels very much like if you were told to write the same sentence over and over and over, that at some point you're going to miss a word just because it's blurring together and it's getting hard. Like if you were trying to read it, read this word 20 times, it's going to be hard to keep track of if you're on 15 or 19 because it's just blurs together.
yes. It's making me think of, I don't know if this has it ever happened to you, dear listener, where you're typing on your keyboard, but you're not looking at like the keys, and unbeknownst to you at some point, your hand shifted one key to the
Yep, yep.
side, and now you're typing things that aren't words because where you expected the O to be, you're hitting P, and your whole right hand is now offset by one.
Yes, exactly. The other way that we typically see mutations caused are mutagens. Mutagens are things that cause mutations.
Yes. So if in my analogy of if a replication error is it's internally caused and it's me, I shifted my hand over one key by accident. The mutagen is like when the cat walks across the keyboard
Yes,
and
yes.
just hits a whole bunch of keys.
Yep. These can be chemicals or physical effects, and they cause mutations in different ways. Three most common things they do is either acting as a base analog that they get mistaken as one of the nucleotides and put in place, or they react directly with DNA, actually have a chemical reaction with the DNA strand and cause some sort of damage, or acting indirectly where they cause the cell to synthesize stuff that damages the DNA, like peroxides or something. With chemical mutagens, the most common ones you'll see are base analogs. These cause point mutations just by making a mistake where some part of the chemical was put down as a base instead of one of the actual nucleotides.
Right. It's a molecule that isn't adenine or whatever, but it's close enough that it slips into the works. And now you have
Exactly.
that's not a proper DNA base pair.
Exactly. Well, it's kind of like how it was. Is it carbon monoxide where that is poisonous because our red blood cells like to grab it more than they like to grab oxygen because it is a similar enough thing.
Yes, I think that's also why cyanide is bad. That
Yeah,
it
right, right,
a
right.
lot of toxic materials are like this because it slots in to like the nerve receptor or whatever where something good and useful is supposed to go. This thing is toxic because it fits in there and is basically a wrench that's plugging up the works.
Yes, that and because that's how proteins work for everyone who's not as familiar. Proteins have a active site, and it is basically the site that they bond to other things with. And the way they know what to bond with is that they are a specific shape, and so they will only bond with the things that are also a fitting shape. The reverse plug, and if something else has the same plug or a very similar plug, a protein might grab the wrong thing and the protein doesn't know anything because it got its plug that it's supposed to grab, but now the thing it grabbed won't do the second step of the job that's supposed to happen, and so that can happen here.
Yep. I've put together IKEA furniture. I know
Yep.
how it goes. Sometimes you're like, wait, where's the screw? It's like, oh, that you put something in a place it wasn't supposed to
Yep.
go.
You use that on step 3 instead of step 6.
Yep.
You can also get deaminating agents. Deamination is the removal of the amino group from one of the base pairs of the DNA. This is something that can happen spontaneously, but the rate can be increased by certain chemicals like nitrous acid and sodium bisulfate and things like that, that deaminate certain groups. They will deaminate certain nucleotides.
Basically, just like stripping a piece of the molecule off.
Exactly. And when this happens, very much like the tautomers, they bond with the wrong base.
Hmm. Sure.
And so adenine will bond with C instead of T, and cytosine will bond with an A instead of a G. So you get the same effects happening that when they get this amino group removed, they now read incorrectly. Similar to that is alkylating agents that add an alkyl group to the nucleotide instead of removing a thing. And these can cause a couple of, there's different versions. There's methylation, which is a version of this, and there's different kinds and they have different effects. But some of them can be very drastic and block replication because they can actually bind the two strands of DNA and not allow them to split.
Oh, so I used the wrench in the works as a
Hmm.
analogy before. This is like gum in the works.
Yes, exactly. You can also get intercalating agents, which are flat molecules that slip between the base pairs and add a gap, a physical
Uh,
gap. And this can result in an insertion mutation because the polymerase comes across more spaces than there should be and puts down more nucleotides than they should.
so basically, a lot of these chemical mutagens, because this is the thing that it's so fun when you get down to this scale, it's just all physical pieces bumping up against each other. And
Yep.
so many of these chemical mutating agents are just pieces that block the path of DNA replication, or break a chunk off of a molecule, or stick a chunk onto a molecule, or slip in between the molecules. It really is just like the conveyor belt system is going, and you're not supposed to put new stuff or remove any pieces or put anything on there that's not supposed to be on there. That's a lot of these chemical mutations. That's basically what's happening.
Exactly. They're messing up with the interaction of the protein machinery that's going to be then working on your DNA. The physical mutations are not as physical as you, in the way we typically think of physical. It's not like you're being hit with something from the outside.
Physical, like physics.
Yes, exactly. Radiation is the
Hmm.
most common form, different kinds of radiation, which is a physical interaction. A particle from an unstable atom launches off and passes through your body and hits part of your DNA and affects it in some way.
Yes, this is it. By the way, we talked about this in episode 167 when we talked about radiometric dating.
Yes.
That radiation is just single, often subatomic particles that are just shooting off of atoms. So it is an
Yes.
actual microscopic little piece of an atom that's getting released, and it can bounce around in the environment and cause problems with things that it crashes into.
Yeah, because they're very high energy, so they go out with some force and can do some damage. UV radiation is extremely common. It comes from the sun. This is why it is so important to protect ourselves from overexposure to UV, is that it can cause mutations and damage to our DNA. One of the most common things it does is it will dimerize parts of your DNA, which dimerization is forming a bond between two identical molecules. And so it acts on base pairs that are the same base pair next to each other. So two C's, two T's, two A's, two G's, and it will bind them and cause a mistake, usually a deletion mutation, because they get read as a single base pair. And so whenever you have situations, evidently it's most effective on thymines,
Okay.
that it will dimerize them and fuse them together. So it's just binding up parts of the DNA as these little particles hit you. Ionization radiation is another very common cause of mutations. This one will have a variety of effects depending on the source of the radiation and its intensity. So, could be point mutation, insertion, deletion, basically all of your typical kinds. Some acting directly on the DNA, others causing side effects in your cell, changing molecules in your cell that then have effects on DNA. But by far the most common of physical source of mutation, which I think will probably surprise many people because I was shocked, is heat.
Yeah, that makes a lot of sense. I think that like UV radiation and radiation in general is a very famous source of mutation because
Yes.
that's what we often think of. You know, it's the classic cliche examples of like this river is next to a power plant and it's leaking radioactive materials and the fish have extra eyes and fins and whatever. That sort of
Yes.
radioactive. But heat, A, is a lot more common and easy to come by than a lot of other forms of radiation. And heat is a form of radiation.
Yes, it is. It is indeed. And heat changes in thermal energy affect the way molecules interact and function and how well they can stay together, the stability of a molecule. That's what happens here is that it can stimulate water induced cleavage of one of the bonds that is a component of a nucleotide. So basically, it can split up particular bond in the nucleotides and cause them to detach from the sugar base that
Hmm.
is the one of the main components, one of the main structures. And this results in what's called an AP, an apurine or apyrimidine baseless site that has lost its base.
Hmm.
Also, it does not occur equally across. It occurs more often with purines than pyrimidines. So this is happening all the time. The sugar base that's left over is unstable and degrades, leaving a gap. This is happening constantly when you are out in hot weather and in hot environments. Roughly, the number I saw noted in the textbook was 10,000 AP sites are generated in each human cell per day.
Each cell.
Yeah.
Wow.
Now you might be saying, how do we survive that? And we'll get to that in just a moment.
Yes. How are we not goo?
Yes, we have systems in place to handle this.
Well, and this, speaking of systems that are in place to handle this, and maybe I'm jumping ahead a little bit, but I think that you are correct in heat immediately thinking of it as a major mutating factor is a bit of a surprise. But thinking about it a little bit longer, the one most sort of famous tangible adaptation that many animals' bodies have for avoiding mutation is most mammals keep the testicles in a little pouch that is separate from the body. The
Yep.
point of that is to keep that the germ cells that are developing in there a little bit cooler than core body temperature. And the
Yes.
reason for keeping them cooler is because heat can cause all of these problems with DNA replication.
Absolutely. So we have systems in place to mitigate this, and we'll go over those in just a moment. But first, let's talk about some of the effects that these mutations that we've discussed can cause.
Yes. Which of the types of mutations that you've described so far gives me claws that come out of
Right?
my knuckles?
Not none of them by themselves. You you need a lot. When we look at the effects mutations have, there are differing degrees to how easy it is to tell what that effect will be. If it is a mutation in a part of the genome that doesn't code for anything, it probably won't have an effect. A bunch of our DNA does not code for proteins, does not do any work in telling the cell what to do on a regular basis, at least. And if those parts are mutated, those mutations will have no noticeable effect on the functioning of the cell or the, you know, outward functioning of that organism. These are often called silent mutations. You did mutate and that mutation stuck around, but it's not having an effect, so it is not actually a noteworthy mutation. Roughly about 98.5% of the human genome could be mutated this way with little effect. So, like, most mutations that set in are not going to be having dramatic effects on that DNA strand.
Right. And this could be because it's part of, like you said, a non coding section of the DNA. But I could also easily imagine that if a mutation happens to a gene in a cell that doesn't use that gene, like if
Yep,
that gene is important for liver cells, but the mutation happens during replication in brain cells or something, that's not going to matter
exactly.
because that gene's not active here.
Yes. If it does have an effect, They often get distinguished between direct and indirect effects, having a direct effect on the way the DNA codes stuff, or the effect it has ends up having effects down the line of the function of that cell or that tissue and is notable in some other less directly connectable way. This is going to be the case if it happens within a coding region of DNA. Typically, these defects are going to be negative because when you go around randomly messing with code, you typically break the code more often than you do anything else to it. In the cases of like point mutations, these can have one of four effects. Synonymous change, which it does change a codon, and that does specify things differently, but it ends up specifying for the same amino acid just with a slightly different code.
Yeah, this is a thing. If you remember, anyone who's, if you took a biology class with, in a, I guess, a similar context, I remember there's a chart of like
Hmm.
what combo of nucleotides makes each amino acid. And some
Yep.
amino acids can be formed from multiple combos. So
Yes.
if you replace one with the other, it doesn't actually change the end result.
Yeah. Functionally a silent mutation. Even though you changed a coding region of DNA, you still are going to build the same amino acid, so it doesn't matter.
It's like typing a capital I instead of a lowercase l. It's like, yes,
Yes.
technically you did, so technically something in there is wrong, but no one's going to notice.
Nope. Everyone's going to read that exactly correctly. A non synonymous change is also called a missense mutation, where the codon was altered, a different amino acid is produced, and so the protein that is coded for and built by that section of DNA will have a single different amino acid in it.
Hmm.
This often also doesn't have much of an effect because most proteins can survive a single mistake like that. Their overall structure is enough to still retain function. It may be slightly less efficient or slightly functioning a little bit differently, but overall it's going to still get the job done that it was built to do, unless this is coding within like a specifically functional, the activation site of the protein or the bonding site of the protein, then it likely won't have a huge effect.
Right. It's like if you put the wrong fender on the car.
Exactly. It's
It's like,
still
yeah,
gonna do. It's
that's
not gonna fit.
wrong, but it's still going to be a car.
Yeah, it fits a little bit weirder, but it you can get where you're going. If it converts the codon, there's two ways it can convert. It could convert a codon that codes for an amino acid into a termination codon. So a codon that says, all right, stop, you finished the protein. Or vice versa. It could change a termination codon into one that codes for amino acid. These will be called a nonsense mutation if it terminates in the middle of a coding region because
Hmm.
you've turned one of the amino acids into a stop signal and the protein only gets half built. Usually, this is very negative on the effect of the protein because you have 2/3 of a protein. This will depend on how much is lost, but usually it's not good. If it converts a termination into a amino acid codon, then you get what's called a read through, and it continues. It starts building a protein and then reads through into the next protein, basically, and you end up getting an extra chunky protein with
Hmm.
extra bits on it. This usually isn't terrible. Most proteins can tolerate a little bit of addition, but the longer that's added, it might start messing up with how the protein folds and how it physically functions. With deletion and insertion mutations, it depends on how much is deleted and inserted. If it's pairs of three, then usually not that big of effect. Because, like having the wrong amino acid, missing one or having an extra usually isn't terrible, unless it's part of one of those critical parts of the protein again. If it's not a multiple of three, then not only did you mess up that one codon and that amino acid, everyone downstream of it is messed up. This will usually ruin the protein or have a major effect. So that will mess up the building of that protein because it is now all slightly wrong. With all of these effects, usually you will see if there is an effect on the protein, the two ways that they often describe it is that there will be some loss of function. Protein is not working as well or not able to do something it did, or a gain of function where the protein is doing something new, something it wouldn't usually do. The gain of function one, like loss of function can be bad because then that cell is not able to do work that it would normally do, which could lead to cell death, or cell underperforming. Gain of function can be worse because this is how you can get to things like cancer. Because
Right.
now, if a cell's doing something it shouldn't have been doing, that's how you can get over replication and taking up too much nutrients and things like that. All of these mutations can also happen in single cell organisms, but it's a little bit different because for them, every mutation that has an effect affects the whole organism because they only have one set of DNA.
Yes, I was just thinking about because you were describing, we're describing these effects. Of, well, if this happens, it can completely ruin the coding of that protein. And that's dramatic, but we're talking about a single protein within a single cell.
Yes.
Even if you get a mutation during replication, that throws a real wrench in the works and it creates an abomination of a protein that interferes with the cell's ability to function and the cell completely collapses and falls apart. That, yeah, but my body has billions of cells.
Yeah. So every time I scratch my skin too hard, I kill some skin cells.
Absolutely. Yeah, the body is constantly developing new cells and destroying old. I mean that are you would lose cells all the time. So
Yes.
that's most of the time these mutations, even when they are a significant negative mutation, are completely inconsequential in the context of our whole multicellular bodies.
Yes.
But if you're a yeast and you are
Hmm.
a single cell organism, then a mutation in your cell is a mutation in you. That's your whole
Yep.
being.
Exactly. So for them, every mutation is like a germline mutation in the fact that this will either end your line or be passed on to every cell you split into. So it's
Yeah.
much more dramatic with single celled organisms. And on the note of like, if bad mutations happen with our cells, usually to get to something like cancer, you need a series of mutations to build up that cell has a mutation, it splits, and its daughter cells have that mutation. One of them mutates again, and
Hmm.
after a dozen steps of built up mutations, you now have a cocktail that is a cancerous cell that is
Yes.
now functioning fundamentally differently than it should.
And I do really like the point that you made that there's loss of function mutations and gain of function mutations. And loss of function sounds very intuitive. It's like, yeah, something's
Yes.
broken and it just doesn't do what it was supposed to do. Gain of function on the surface sounds like a good thing.
Yeah, superpowers.
This cell has a superpower. It can do something it wasn't supposed to be doing before. But again, we are multicellular organisms. So a cell doing things it's not supposed to do is oftentimes not good. That is
Yes.
like a car on the highway that just starts playing by its own rules.
Yep.
Yes, I'm glad that that person has freedom to do whatever they want, but they're going to cause chaos on the highway. And that is when you
Exactly.
get stuff like cancer. That's a cell doing something it's not normally supposed to be able to do.
Yes. So it's the side effects very much depend on the context of where it's happening and which cell it's happening with. With microbes, there are also some very often seen results that also can be seen as loss or gain of function, but typically are grouped into more specific situations. One is that a mutated cell can become an auxotroph, which is a cell that can now only grow with some nutrient that was not previously required. So it now has more specific needs than it previously did. There's also conditional lethal mutations, which are that in certain situations, the cells malfunction, where the
Mm,
mutation isn't apparent under normal circumstances, but then in like higher heat or lower temperature, this mutation becomes apparent and the cells start to malfunction or die. Regulatory mutants have defects in promoters and other regulatory things in their bodies and can express things that are normally switched on and off, but are now always switched on or always switched off or not switching correctly.
hmm.
And then once again, they can have things that have no effects or that might give a positive effect, but we'll get into that later in the discussion. To wrap up this first section, after all of that scary talk of all the negative things that can happen to your DNA, let's talk about why you aren't turning into a pile of goo or growing extra buds at a moment's notice
Right.
every day.
I was taught not 10 minutes ago that
Yes.
every single one of my cells has 10,000 heat induced mutations every day.
Yep. There are multiple stages and ways that our cells fix mistakes and repair damage to the DNA. This is a like many redundancy system in our body that means that even though we are experiencing a huge amount of potential damages and mistakes in replication to our DNA at any given moment all the time, only a very small percentage actually make it through to being a permanent mutation. So all of those things were the ways mutations can happen, and even though all of those are happening fairly regularly, most do not become mutations. Most end up being repaired, fixed, and go back to being the DNA they should be. This has led some to say that a definition for mutation could be a deficiency in DNA repair. That really, mutation happens when the repair is not measuring up or up to snuff. And so there's a very important relationship between these two. And that they've noted that many like genetic disorders in humans are the result of reduced DNA repair systems. And
Interesting.
so this is a very important and like this is important to life being life, because without this, our DNA would have just been torn to shreds by the environment of Earth and the
Yes.
mistakes of chemical reactions in a short time. So without these, life would not persist. One of them is DNA polymerase itself has a system for checking as it lays down the new DNA strand. The two main ways is that A, polymerase has the selection process for selecting nucleotides, which is, has ways to be more accurate and does a still fairly decent job, even if it has a 5 to 10% error rate. And it checks whether it's grabbed the correct one at multiple stages of laying it down. And then there is a way for it, when one does get put down, for it to immediately be kicked back out and the correct one put down. This is often called proofreading, even though it's not actually proofreading. There's two features to polymerase: the polymerase function and an exonuclease function. And these functions are constantly fighting for balance. And typically the polymerase wins out, and that's it laying down nucleotides. And its function is heightened when base pairs are matched. That chemical situation heightens polymerase action. So when base pairs are not matched, polymerase function dips. And during that dipping, the exonuclease function pops up and will kick out the unmatched nucleotide.
Yeah. All of this is passive activity of interacting molecules. And
Yes.
so it really is exquisite in its simplicity of each of the individual pieces, where
Yep.
it's like, you don't have a robot that's making all these checks. It's like saying, all right, this piece has to fall down to the bottom of this contraption. We're going to put there's going to be a series of different obstacles in the way, such that only the correct shape of piece should be able to make it past all of these obstacles.
Yes.
And then if the wrong one gets in, the whole molecule just sort of breaks a little bit. And when it breaks, part of the process of breaking can kick out the thing that wasn't supposed to be there.
Yes.
That the molecule isn't functioning properly, and one of the symptoms of it not functioning properly is this other enzyme becoming active, which removes the thing that is plugging up the works, and then you go back
Exactly.
to. And so it is this very, we're talking about this sort of brief overview because we are not geneticists, and putting it in the very easy colloquial descriptions of like, it's proofread, and these are terms that geneticists use, right?
Yes.
Proofreading and checking and performing repair, but it's all just a Rube Goldberg machine where the different pieces are refined enough that it keeps itself moving.
Exactly. And so, yeah, because your cell doesn't know what its DNA is supposed to say. Because your cell doesn't know anything. It doesn't have memory and thought, but it has ways to recognize errors based on the way its systems work. And that when something does not pair up the way the system of our cells are meant to work with it, there are things that step in to set it back to the way that works. Those steps are things that avoid mistakes getting put into the DNA. If a mistake does get put in, we also have ways to repair those mistakes. These repair systems, there's a number of different kinds, and they work on slightly different kinds of repair and repair them in slightly different ways. We have direct repair systems that note a mismatch and fix that mismatch, kicking out the wrong one, putting in the correct one. This is actually a very small part of our repair system. Some of the more active ones are things like excision repair, which will cut out a section of mismatched DNA and lay down the correct section. And they do this in a couple of different ways. A similar one to that, which is mismatch repair, also can kick out, excise a stretch, and then repair that resulting gap. Neat thing about the way this one works is a question could come up of, but how does it recognize which half is the part that needs to be repaired? Like, how does it not know that the other side is not wrong and the incorrect ones are supposed to be there? When your DNA gets replicated, DNA has what's called a methyl group. It's methylated. That takes some time to happen. So the newest laid down strain has not yet methylated. So it only acts on the unmethylated portion of the DNA to kick out the most recently added nucleotides. And then we also have repair systems for recombination where things have gotten shifted in your DNA for those more dramatic things that can happen, even up to a piece of DNA being cut, like fully both strands cut. We have systems that can reconnect those and reestablish a break in DNA. We also have repair systems that act before replication. So some of these might be acting shortly before replication happens to go catch any mistakes before they could be replicated wrong. Others happen right after replication to try to catch newer mistakes that have just set in. So this is happening kind of constantly and is a non-stop way to try to catch as many mistakes as possible. So in all, a very few amount of mutations, a small percentage, actually get fixed into the genome and make it as a new coding to that DNA.
Yeah, and then it's important to remember that if a mutation does make it in there and does become active and does affect the functioning of a cell or something, there are also cellular mechanisms
Yep.
for just, apoptosis is a feature, a function built into all of our cells, which is called programmed cell death, which
Yes.
effectively, if a cell isn't functioning properly and it is beyond repair, it just destroys itself.
Self-destructs.
Self destructs, it comes apart, it re delivers all of its component pieces back into the body to be recycled and used again. So even if you do get a mutated cell, those cells often don't get the chance to cause any major havoc because they die off.
Yes. And if one does get damaged in a way that is too much for the repair system to handle, like a section gets too mutated or too messed up, there are systems in place, at least in certain organisms and cells, that when it's time to replicate that DNA, they have systems that will skip over overly damaged sections
Mm-hmm.
and not replicate that section. They noted that E. coli is known for this, that basically when a cell has too much damaged DNA, the two options are die, don't replicate, or figure out a way around that damage. And E. coli goes with the second option and has ways to skip over major damage, and still replicate the rest of the DNA, which will probably have a negative effect on those resulting cells, but they will be able to replicate.
Yes, they will survive.
And so there's even a escape pod mode of cut off the damaged areas and we're going on without it.
Yes. And this is much more obvious when we're talking about single-celled organisms. But even with multicellular organisms, if you imagine the scenario where a mutation slipped past all the repair mechanisms, the mutated cell doesn't get destroyed and becomes active, spreads throughout the body, and ends up creating a harmful circumstance within that whole body, that body doesn't often get the chance to have a negative impact on the whole population or the whole community because that individual won't survive.
Exactly.
And this is sort of the last line
Mm-hmm.
against the spread of harmful mutations, which is just if the body doesn't function, that individual dies and doesn't pass along that harmful change.
Exactly. It does not get to spread among the community and increase in number. For those mutations that do make it through, that don't end the life of the individual, and in us, multicellular organisms are in a germ cell, those are the drivers of evolutionary change, which we will discuss after the break. How these mutations affect evolution and how we track them when studying them. The connection between evolution and mutations is almost one to one, in that it has been said, and you could say, that every genetic feature we see in life on Earth at one point was the result of a mutation. So, all the diversity of life on Earth is the result of mutation. Now, not a mutation alone, there are other factors, natural selection, and survival rates, and different things that select for and against those different mutations. But the source of that diversity initially comes from something changing in the DNA, and that change sticking around and affecting that life down the line and through its lineage. We already talked about some of these side effects that mutations can have, loss of function and gain of function. Typically, these are not good losses or gains, because taking away, making something that did a job in your body do less of a job, usually not going to be helpful, and making it do something randomly new, also usually not going to be helpful. But every now and then, one of those gain of functions and potentially a loss of function can be beneficial to the survival of that organism, the functioning of their cells and how they interact with the environment around them. And these things that increase the fitness, the survivability, or the reproductive success of that organism are what we often describe as adaptations. Those changes become adaptations.
Yeah, it's the link between mutations and evolution is often misrepresented in a very dramatic fashion, where we imagine this scenario of an organism has offspring and the offspring has a mutation, and that mutation is that it has a longer neck or that it has
Mm-hmm.
claws or something tangible, and it has a new superpower like the X-Men. But in reality, most of the time, what we're talking about with mutations, and in the first section, going over just the incredible diversity of ways mutations can happen, things that can cause mutations, ways that the body and proteins in our cells can respond to mutations. What all of this does is it just means that every new cell is a little bit different combination than what came before. What
Yes.
mutation does more so than introducing some fancy new thing is just varies up what features and functions are available in a community or in a population for evolution for natural selection to happen, right? That
Yes.
natural selection is all about comparing and contrasting. Something survives better than something else, and that exists because of this diversity. What mutation really does at the core of it is it just adds a bunch of tiny variations across every cell and every organism. And those little variations are the foundations for natural selection to end up leaning more in one direction or another.
Precisely. And sometimes it can lead to very clear cut advantages of, yeah, these bacterias got a couple of mutations and are now immune to the antibiotics that usually
Right.
would have killed them. That colony's probably going to do better than its neighbors who didn't mutate that way because there is a single deciding factor that they have now gotten around via mutation. But often it will just be adding variation to a population. And this variation gets selected for or against by natural selection, but that doesn't always mean that it will be evenly selected. Sometimes it's good to just have a 30% that have this feature and 30% that have this feature, because then when a drought comes along, enough of your population makes it through because you had this 30%. But when a famine comes along, that drought 30% aren't good for that. So you want some variety. This genetic diversity is the foundation, is why mutations are so key to evolution happening. Remember again, only a small amount of mutations are making it through the selection process of our cells, and in us multicellulars, only those in germ cells, only those in the reproductive parts of the body are going to be able to be passed on and become a inheritable mutation. There's also multiple ways this variety of genetics can be affected in can show itself in evolution. Typically, our go-to example is how it affects natural selection, that mutations introduce a variety of phenotypes of organisms functioning in slightly different ways, and now the natural pressures of their environment select for or against those new varieties. And this is why negative mutations still play a role in natural selection, because that is now a genetic disorder. If you're born and you lack something that was key to surviving some situation, natural selection selects against that mutation and it is taken out of the gene pool. And then positive ones that infer some advantage or better adaptation to a situation get selected for. Those animals or organisms are more successful and breed more successfully, and that mutation spreads through the gene pool, becomes more numerous. There are now more individuals who have that mutation, and they can then multiply it again when they reproduce. And eventually, that could become a set mutation for the whole species, as that is selected for, and those without it get slowly selected against. And now you're all, redheads or whatever the advantage was. Yeah, I said red hair because that's a famous like mutation that gets pointed out in humans of like, yeah,
Yes.
that's the result of a mutation.
Or like lactose tolerance,
Yes, exactly.
which is one of those very famous examples of a well-known mutation that
Mm-hmm.
happened in multiple times in human populations, where we can follow the trajectory of it and go, here, this is it, this is the part of the genome that got tweaked just a little bit, and what that did is it turned off the thing that turns off our ability to process milk.
Yes.
There's a switch that's supposed to turn off, and a mutation broke the switch. And because that switch is broken, I can drink milk.
Yep. And in most human communities, that switch is still active. But
Mm-hmm.
in the communities that were relying more heavily on milk and milk byproducts, that mutation was an advantage and spread among those communities to where, in those groups of humans, it is almost 100% because it was so useful.
Yes, because at some point, thousands of years ago, Europeans were sitting around drinking milk and eating cheese and stuff, and a few of those people weren't having horrible intestinal issues,
Yes.
and they were far more capable.
Yeah, they were much happier and
Yes,
able to go on.
they were able to raise their kids.
Yes. Yep. So that is the typical way we think of mutation affecting evolution. But mutation also can lead to what's known as genetic drift or neutral selection, neutral evolution, which is just as mutations happen and build up, the genome and the gene pool of individuals and species just slowly meanders and just drifts apart from each other and just becomes a bit more random. And this is notable. This is important to mention because for a long time, genetic drift was kind of looked at as, yeah, it happens, but it's not important. Natural selection is the important part. But as more research and understanding into the effects of mutations has gone on, we've realized that no, genetic drift can be quite dramatic and is a constant presence. So it is a major player in the evolution of life on the planet. Genetic drift can have similar effects in that certain traits can become fixed just due to random chance. That so happens this trait got spread, and now basically everyone has it. So this is now a trait of your species. It wasn't selected for or against, but because it wasn't selected against, it also wasn't pulled out of the population and it just happened to get spread. It can also cause the deletion of traits through the same thing. If it's not selected for, there's also nothing stopping it from slowly disappearing. That's why we talk about vestigial structures, that if there's no selective pressure on keeping this the shape it is, then it can just slowly get more mutations and become more random in its shape and eventually maybe disappear.
Yes, again, it contributing variation.
Exactly. The one thing that genetic drift functions notably differently than selection on is that it really reacts differently to the size of the population. In
Mhm.
a large population, you hear this called the law of large numbers. With a large population, there's enough interbreeding and enough variety that the drifting gets kind of kept under control because there's the population's too big for the entire population to randomly drift in a similar direction, so they are all keeping each other's drift by interbreeding with each other in check. In a small population, you do have small enough of a number that drifting can kind of shift the whole population in ways more dramatically. This is often noted in things like the founder effect in bottlenecks, where a population gets reduced suddenly, either due to a famine that kills off a whole bunch and now you have a small population, or founder effect where a only small group migrated to a new land or made it to an island. And in those small gene pools, genetic drift can be much more dramatic and you can see more intense changes just due to the random mutations happening.
Yes, there are a few famous examples of this with human populations. I
Mm-hmm.
think one of the first famous ones that I learned about was here in the U.S. with certain Amish communities, where Amish communities tend to marry and have children within their own community, which
Yes.
means that whatever mutations or genetic quirks were present in the earliest members of the founding members of those communities, those have become widespread. And it means that certain diseases or certain susceptibilities are particularly common within Amish communities because they started out with that particular variation in the gene pool. I think the same thing is true, for example, of certain Orthodox Jewish communities.
Mm-hmm. Mm-hmm.
Anytime you have a group when we talk about it in nature, it's often this is a group of animals that the area flooded and now they're stuck on an island and they can't escape. It
Yes.
happens with humans often with communities that voluntarily isolate themselves
Yep.
by reproducing, right? Having children within the community, you limit the expanse of that gene pool. And so you can get what would otherwise have been relatively rare variations become very common within that community.
This is why you can get, communities and nations that have a single hair color as the predominant hair color or eye color, because at some point it was settled. That area was, migrated to and settled by a group of people. And if, 7 out of 10 of them were all blonde, that became your base set of DNA. And as they multiplied into an entire nation of people, now 80% are blonde because that's the DNA you started with set that. with these small populations, mutations also have a stronger effect on the changes you'll see within that community and within that new population. So the randomness of mutation also itself is just a lone driver of evolution. No selection, just random change can over time cause major shifts in species. And this is why if we fast forward a million years into the future, humans will look a little bit different. We probably won't look crazy different, but the proportions of genetics will have just shifted over time because we are constantly changing. Even if we're not being selected for, even if we're avoiding natural selection, like us humans have done everything we can to remove ourselves from the natural selection process with hospitals and safety and medical education, all that good stuff, we're still changing. The genes that if I were to have had children that I pass on to them would have been slightly different than the genes my parents gave me. Genetics would have shifted. Another very cool way that mutations can affect evolution is in convergent evolution, which is, we've talked about that here on the podcast a ton because convergent evolution is arguably the most fun example of evolution to look at because it's just so quirky and cool of when two organisms evolve very similar answers to the same ecological pressures. We both evolved to be shaped streamlined for swimming in the ocean. We both evolved our front limbs into wings to be able to maneuver more, well into the air, et cetera, et cetera. Sometimes when we see convergent evolution examples, usually, I should say, when we see those, they're evolved in very different ways. You came to similar answers, but they're not the exact same bones. They're not the exact same structures being used. The mutations that caused that and led to that are not going to be the same, but sometimes they are.
Yes, yeah, it's most of the time convergent evolution is like two different communities that both invented boats. And it's
Yes.
like the shape of your boat is different, the wood you use to build it is different, the steps you use to build it are different. The end result is a thing that floats on water and can carry you across the river. But
Yeah.
every step you took to get there was fundamentally different. Sometimes, though, you do get this in the case of what we're, with mutations. Sometimes you get it where it's like, no, these two people came up with darn near the exact same idea with the exact same materials and made basically the exact same boat. Sometimes the actual genetic changes are also convergent.
Yes, the cellular action that became this convergent adaptation was a mutation in the same genes in both groups that happened separately. Not a mutation that was passed down to both, but the same random changes in damage or mistakes caused a change in the same gene that led to the same advantage. There are numerous examples of these, but one I found just to give a actual example is a study that was looking at the genetics of deep sea fish found that many of them had a mutation in the gene called RTF1, which helps control how DNA is read and expressed, so the functioning of DNA in the cell, and that this mutation seems to have influenced the efficiency of this gene and likely is having effect on them since they're living in such an extreme environment, extreme temperatures, extreme ecosystems, and everything about it unusual to where most fish live, and that this seems to have led to some advantage, and that this same mutation in this same gene has happened at least 9 times in deep sea fish.
Ooh, 9 times.
Yes.
That's very cool.
And so it is fun because once again, when we're talking about evolution, we're dealing with large, huge populations of individuals, each of them with many cells full of DNA. So even though that is a seemingly astronomically unlikely chance for that same mutation to have happened, this is happening constantly. Mutation is
Hmm.
happening constantly. The cells are reproducing constantly. The animals are reproducing constantly. So all you need is enough individuals to get the same mutation on the same part, and it's going to be advantageous every time it happens in a deep sea fish. And so that
Yeah.
advantage will immediately be there.
Well, and like we mentioned before, certain mutations are more likely than others. And
Yes.
so if this is a mutation that happens, you mentioned before that certain mutations happen more to certain base pairs than to other, right? This
Yep.
is
Yep.
more likely to happen to thymine than it is to happen to cytosine or whatever. Well, if this is a mutation, it's one of those mutations that happens to thymine, well, then that means that it was slightly less unlikely
Yes,
to have happened.
exactly.
And if it's caused by some sort of, right, if it's caused by low temperatures, make it more likely for this kind of mutation to happen. Well, if you're living in the deep sea, you're in a similar circumstance. So again, there are random components to mutations, but there are patterns to what mutations happen when and how and where. And so it isn't nearly as unlikely as it might seem at the face of it.
Yes, I found one metric that I think really captures this concept, that even at a very small, percentage and chance, when you scale things up to the equation of generations and, in paleontology, millions of years of generational evolution, those small numbers can become quite big very quickly. I found one that was saying that E. coli, common bacteria used in many experiments, on average has a mutation rate of about 1 per 10 to the ninth power, which is 10 billion nucleotides. So per billion, typically there's one mutation on average. So every time an E. coli divides, each of its daughter cells has on average 0.0084 new mutations. So a little bit less than 1% of their DNA
Yes.
is mutated. E. coli divides very quickly. They can divide up to twice an hour, which means that within a 10-hour period, a single E. coli could become a million cells. And 1% of a million cells is 10,000 E. coli.
Yes.
So these mutations don't have to be happening on some comic book level scale for really dramatic shifts to happen via evolution. Just the slow accumulation and the random chance, there's enough individuals that random, it's kind of like the concept of an infinite universe, that if the universe is infinite, there is, like, the chances are high, but functionally there is another planet out there exactly like Earth, because you're dealing with infinity. All chances have a chance to play out. So even a 0.00001% chance of something with infinity is effectively absolute. But when you're dealing with hundreds, thousands, millions of individuals reproducing multiple generations, that chance of a single mutation having a specific effect actually rises pretty significantly.
And this is also, getting into the broader topic of evolution. And, we often think of mutation as the random effect on natural selection.
Hmm.
And while mutations can be random, right, when
Yes.
they happen, why they happen, how they happen, there are patterns, but, it's not always predictable. How those mutations will then affect the organism is often not random,
No.
right? You might get a completely random mutation that leads to excess hair growth, but predicting how excess hair growth is going to affect the survivability of the organism is actually extremely straightforward.
Yep.
Right? That part's not random. The fact that that helped that organism to survive better is not at all random. The filter isn't guided, but it is highly predictable.
Yes. And as we mentioned earlier, multiple mutations could give you similar adaptation, similar phenotype changes. So the randomness actually becomes a lot less daunting as you look at it at the correct scale and under these circumstances. Now, when we're talking, here we are a paleo podcast, when we're talking about evolution and mutations, we run into a challenge of, I don't have the DNA of past extinct animals usually. So how do we study the path of mutations? The origins and tracing mutations along a lineage or a group's evolution. This is a common issue both in studying past evolution and studying modern life. When we look at mutations like within our body or the body of an organism, tracking cellular mutations within somatic cells is also important because that's important to tracking the development of cancer or the development of other genetic issues within tissues. And so there are systems trying to find ways that can follow the course, quote unquote, of a mutation through the lineage of tissue cells. Because even within our body, the cells have a lineage. My muscle cell number 278, when it splits, will make muscle cell 279 and 280, and then those will go. So there is a lineage. The cells in my muscle are descended from the original cells in my muscle. And so if one of my cells when I was 12 had a mutation that stuck, every cell that's descended from that muscle cell has that mutation. So my muscle could be 78% that original cell now and have this new mutation. So tracking them within the body is also important. I found a couple of papers talking about ways we try to do this in things like model organisms that we use to study stuff like this, like bacteria. We can just track every generation
Yep.
and do the DNA sequences much more easily. But doing full DNA sequences for multiple body cells of a more complex organism like humans, I say as a human, is much more time consuming, expensive, and less feasible. Some techniques use things that directly detect, like using chemical markers that will
Hmm.
mark the mutation and let you mark it across the cells, looking for specific variants in the genetics and using that as a marker. But I found one that said that you can also use mitochondrial DNA and RNA because those are more numerous, like those are more common in the body, and they get more mutations faster than our genome DNA. So
Sure.
they can be much more effective at finding and tracking mutation paths throughout a body. When it comes to studying an ancient mutation, that gets a bit more complex. I found a couple of examples that I thought were good ones to explore this with. One is a study that found two mutations being key to the way our human bodies process hormones like estrogen, testosterone, and cortisol. That they found a set of mutations that allowed ourselves to process and recognize more hormones than just estrogen, setting up ancestrally our ability to have the complex hormonal systems and reproductive systems that we now have. This is a family of proteins called steroid hormone receptors that can recognize different hormones and control the effects of different hormones. So they're what let those hormones have an effect on our biology. Without these receptors, our cells would not recognize these chemicals and we wouldn't, develop differently based on estrogen or testosterone, or be able to use the pain receptor of cortisol and stuff. The ancestor only recognized estrogen, and its descendants gained the ability to recognize more. They used what's called gene resurrection to model what the ancestral gene would have looked at like. They do this by using computational methods. They work off the tree of life of the group they're looking at, in this case, us humans, and then they take a database of the present diversity of that type of receptor and closely related receptors, and looking at the shape of all of these various proteins and the amino acid sequences, the genes that would code for those different ones, we can calculate what the most likely structure of the ancestral form would be for this diversity. And then they can test that reconstructed ancestral gene and protein by synthesizing it in the lab and then testing how that protein worked to see if it works and does what they would expect it to do. And then they can start testing mutations to see what mutations would change that to the ones we now see and identify the most likely mutations that caused a shift from the ancestral state to the current state.
Very cool.
In this case, they found two mutations seemed to be the cause, and that they caused a 70,000 fold shift in preferences away from estrogens toward other steroid hormones. So, this is also a interesting thing to note because sometimes just a couple of mutations in a very key protein can have dramatic side effects.
Yes. Yeah, this is another one of those things that often can be easy to misunderstand is the idea that, oh, well, you changed one thing and it changed a gene and now this whole new function has developed. But often it is like the example with lactose tolerance that we were talking about, which
Hmm.
I think lactose tolerance has been I think it's a point mutation. I think it is a
I think so.
single, at the very least, it is a relatively small, discrete mutation, but that mutation didn't happen in like the gene for cheese eating. It
Yes.
happened in part of the genetic code for the switch that controls the entire process of the body processing milk, lactase in particular, lactose. So you don't need to mutate the lactose gene. If you
Yes.
mutate the gene that controls the regulation and production of lactase, then your single little mutation has caused a major shift to this whole process.
The analogy that just popped into my brain is that this single mutation had a big effect not because it suddenly turned on the ability to drink milk. We were already able to drink milk as infants. It turned off us stopping that ability. So it's like in Fantasia when Mickey animates the brooms. That is, that's crazy magic, but just stopping to when you don't tell the brooms to stop, that's when then the dramatic thing happens.
That's the problem. Well, it's a similar thing that has happened. We've talked about this with things like snake venom, where it's like you didn't need to evolve whole new systems and whole new molecules to create venom. In many cases, all that had to happen was a system that was already present in the body just got moved somewhere else, got
Yep.
activated in a place it wasn't active before, and now it can be utilized for this different function.
And that's that recombination we mentioned earlier in the episode where you can just get the same job, make acid. Our body has a gene for make acid because we do that in our stomach. If that gets shifted somewhere else, now I might suddenly be able to, some other cells be able to make acid, and that could lead to something crazy.
Now you have xenomorph blood.
Yep. So for everyone on the Discord that was talking about that, there you go. Um, Another great example of this, which I had to use because it is a gene reconstruction going back a billion years to the origins of multicellular life. They found that a single mutation set up a system in cells that would allow for multicellularity. So, in a multicellular body, our cells are now parts of tissues and they need to replicate correctly within that tissue. So like skin cells need to replicate in line with the skin. If they replicate up, I now have a random bump on my skin. And
Right.
that's not how the skin is supposed to be shaped. They need to replicate in line with the cells around them. And the way they do this is with the orientation of what's called the meiotic spindle, and the meiotic
Mhm.
spindle is the thing that, when a cell replicates, pulls the chromosomes to either side of the cell. The new, doubled chromosomes pulls them to either side so that the cell can pinch off into two new cells. This spindle, the direction it pulls the chromosomes in, is the direction they are going to replicate in. So the spindle has to be orientated to the cells around it. And it does this via a couple of other cells that act as anchors and one that acts as basically what they described as a molecular carabiner and connects the spindles to the anchor proteins. And this is a protein known as GKPID. This protein is related to another one that is an enzyme that is glutamate kinase, which is just GK. They looked at these two and did a similar ancestral reconstruction of looking at their structures, other similar proteins, to try to figure out what the ancestral form would be like. The GK enzyme is one that is universal to life. It's important in making components of DNA, while the GKPID is only found in animals mainly, and they're our closest relatives, multicellular animal type organisms. Based on their reconstruction, they found what the most likely ancestral form was, and then tested to see what mutations would cause it to shift from the enzyme to protein spindle or carabiner job, and found that a gene duplication in the GK enzyme was one of the first things that happened, and then it began to diverge. When they tested, when they chemically synthesized this, they got the enzyme with the ancestral gene, and then when they did their mutations, they inserted it into insect cells that had their carabiners turned off, and this one worked as the carabiner. So
Cool.
this reconstruction strategy and then testing out what you get from those computed most likely answers in the lab can allow us to track truly ancient mutations. And neither of these are, this isn't the gene for being multicellular, and that previous one wasn't the gene to having, estrogen and testosterone affect our puberty. But without those, our bodies wouldn't be able to detect testosterone, and cells wouldn't be able to grow in multicellular. So these were important steps onto the path that now we see ourselves today. Another way we study mutations in the past, and probably the most common one that comes up, is studying mutation rate and using it to infer evolutionary timings and paths of organisms. Mutation rate is exactly what it sounds like. It is the rate at which mutation happens. Straightforward and simple. Different organisms have different mutation rates. They can be surprisingly low to surprisingly high. The two examples I saw was, once again, E. coli, and some bacteria have mutation rates of 1 per billion or 100 million, so very low, while certain genes in humans can actually have very high, of 1 per 100 to 1,000 base pairs that have very uniquely high mutation rates. This is specifically in a group that codes for different immunoglobulins in humans, and is likely part of the reason we have such a variety of those. There are instances that we found organisms that have surprisingly low mutation rates, and this is where sometimes that term living fossil will get thrown in. And this is indeed correct that we do see some groups that have notably lower mutation rates than the average member of their overall class, their other related groups. And it does not mean they aren't evolving, but they are showing evolutionary change more slowly than an average species would. Famous one that came up in the news, I think this was a few years ago, was that gars were found to have potentially the slowest mutation rate that's been noted in animals thus far. Gars are freshwater fish, alligator gar, long-nosed gar, very prehistoric looking fish, which is one reason they get noted. Part of why that is the case is because when we look at fossil gar, they look very similar, if not identical, to some species of modern gar. And studies have found that they do seem to have a extremely slow mutation rate, which potentially is answering for why physically some fossils are indistinguishable. Like, you would not actually be able to definitively distinguish between the two species if just looking at them physically. When they compared the genome of gars to 471 jawed vertebrate species, the gars DNA seemed to evolve at a rate 3 orders of magnitude more slowly than the other major groups. And in the paper, they note that this is really the only species we've ever been able to scientifically identify that could arguably fit the term living fossil,
Mm-hmm.
because it is actually evolving so slowly that it might be indistinguishable from some of its fossil members. So you do get situations where mutation rate can be extremely low, so much so that it is affecting their evolution. But mutation rates are not a set static rate. You can get variations in rate within a lineage in different parts of their evolution or different situations. Most notably, there are many instances with studies on bacteria noting that their mutation rate seems to increase in situations where they are under stress from their environment. That
Yeah.
they seem to effectively, big quotes on these words, but if we generalize, evolve more aggressively via increased mutation when a situation is not desirable and is stressing their population out. These have been noted to correlate with things called hypermutator alleles, things that increase spontaneous mutation rates within the genome. And so the term hypermutation has come up with groups like this. Also, in regards to that section of human DNA that seems to code for immunoglobulin proteins that mutates more often than other sections of our DNA, there do seem to be situations where organisms have systems in place to increase mutation rate for certain things, certain traits like the immunoglobulins, or under certain circumstances where the need to find a new adaptation to the current situation is critical to survival. This has been seen in scientific studies with E. coli, the one that studied different populations of E. coli in different habitats where the resources were refreshed at different rates, and noted that in the more stressful habitats, after 1,000 days of E. coli evolution, the mutation rates did respond to become more rapid in those groups as quickly as after 59 generations.
Wow. That's very interesting. That
Yep.
altering, and I feel like maybe we talked about this in the news at some point, but that altering the pace of evolution can itself be an adaptive evolutionary trait.
Yes.
Very cool.
Yep. So yeah, like in those sci-fi things, there's tons of examples like the Zerg and the Tyranids and
Hmm.
other examples where it's like these are hyper evolving, and usually it's still goofy in the way it's actually being portrayed, but that concept is not actually 100% sci-fi. You can increase your evolutionary rate. One of my favorite examples that I learned was in our case with the immunoglobulins. It's a set of genes that are called V gene segments, and in these V gene segments, when a mutation occurs, the repair system that comes in behind them instead repairs the original nucleotide to now match the wrong nucleotide. So it, instead of repairing the mutation, sets the mutation as a permanent change.
Huh.
Yeah, the repair systems are different in for those gene segments that are coding for those proteins.
Interesting.
And this then gives us a wider variety of those proteins to make. The most extreme version of this that I found, which also seems to be the most controversial, as in that people have questioned it, but it does seem that it has had support from later studies, is a study that looked at E. coli and once again put them into a medium where they were going to be stressed. These E. coli all had a mutation in their ability to process lactose. So they could not eat lactose as a carbon source. They then were put in petri dishes where lactose was the only source of carbon. So the only way they would survive is if a mutation happened again in that gene to reverse the one the lab had done. And they found that that did indeed happen. But what was most interesting is that when they looked at the E. coli after the fact, it seemed that mutations had happened there more frequently than they expected for the normal rate. It seemed that they
Yeah.
were happening, they were getting mutations in this key part of the DNA where it would need to happen more frequently than should have been happening. So that not only could they seem to change their mutation rate, but target the mutation to sections of DNA where it would be the most useful.
Yeah. Which I wonder if it is that they are reacting to the presence of this molecule in the environment, or
Mm-hmm.
that's a very interesting. It makes sense that
Yeah,
if you can have a mechanism for changing the mutation rate that provides variation that can then be acted upon by natural selection, that seems like a great strategy.
exactly. Like I said, when this study came out, I saw it noted that it was questioned because
Sure.
that was not something we thought was possible for you
Yes.
to target mutation. But other studies with other bacteria seem to have supported it. So it does seem like there is some sort of mechanism for programmed mutations
Yeah.
in at least certain kinds of bacteria. All of this leads us to what is the most paleontology part of the episode, which is that the study of mutation rates allows us paleontologists to use something we love, which is molecular clocks.
Yes.
And we have talked about molecular clocks so much here on the podcast. This is an extremely go to tool for studying the timing and distance between like two species and when they split or when a trait was evolved. This is how we can estimate. Whenever you hear us say based on molecular clocks, it's likely that their ancestor existed this many millions of years ago. This is how we're doing that, is looking at mutation rate and applying it to the evolutionary history of multiple groups usually.
Yeah, so we might take, for example, humans and chimps, compare our DNA, identify what degree of difference there is between our two species' DNA on average, look at what we know of typical mutation rates in apes or other primates, and then say, all right, given everything we know, this is how quickly we would expect mutations to be building up on average. This is how much difference there is between our lineages. Let's run the numbers. How long would it take at this rate of mutation for that much difference to build up? And we get 6 or 7 million years, or whatever the answer is for whatever circumstance.
Exactly. So basically, we're working off of the concept that as mutations happen, they happen in a rate, and over millions of years, that rate will be fairly consistent. It'll average out to a fairly consistent rate, and we can use that as a pacing mechanism to track the evolution of different groups. To do this, we have to do what's called calibrating the clock.
Mm-hmm.
So if we want to say, all right, we've got these two species, when did they likely split apart from each other? Well, we have to calibrate the clock. So what we would do is we would look at, if we're dealing with modern things like bacteria or human populations, then we can just do the genetic study and time everything, and it's very easy. But with most groups, you will have to use the fossil record to supplement the genetic data and time the divergences. So you would look at the group. If we're looking at you know two species of bird, don't know what their ancestor is or where it was, but the species next to one of them, we do have a fossil of their ancestor. So the ancestor becomes the most recent they possibly could have diverged by, the oldest ancestor we have. We look at the genetics of those two and we look at what percentage are they different. If they are 5% different, then I take the timing. If the ancestor was 5 million years ago, then over the course of 5 million years, 5% of difference happened, which means a mutation rate of 2.5 per 5 million years. Because you have to split, because they each
Mm-hmm.
were evolving a portion of that 5%. And now I have my mutation rate for this group. And if it seems like that is a reasonable rate for the other two, we can calculate. So you have to have one example to calculate off of, and then you use that math to apply it to the situation you don't know.
Sure. You can also, there's a lot of research looking into studying the genome, like you mentioned with GAR, with alligator GAR,
Mm-hmm.
examining the genome among populations of living species to estimate the mutation rate generally, an average mutation rate, which we can then compare with the results of other, right? If we estimate a mutation rate and it is wildly different from every other known bird mutation rate, we can say, all right,
Mm-hmm.
let's reassess, let's double check.
Yes.
But by putting as many of these pieces together as we can, comparing fossil data, genetic divergence, and mutation rates, we can estimate how long and a certain amount of change took to take place.
Yes. And you need those multiple parts because like just looking at two groups and seeing how genetically different they are won't give me a mutation rate unless I know how long they've been evolving separate from each other.
Right.
You have to have these multiple ways to get the full picture of how different are they, how long ago did they start being different, and how much change does that then tell us they were changing over time. There are different ways to calibrate. Some one called node calibration is about timing the splits of branches in a phylogenetic tree. So when you place all of an organisms on a phylogenetic tree, the nodes are where branches branch off from each other. And that's very much what I described of you placing the oldest fossil at a node, and that becomes the plate, the pin that holds down that node in time, and then you go from there. Tip calibration is one that seems to be a bit more flexible and treats each fossil as a taxa, and you put those at the tips of the branches, and then comparing molecular data from your modern groups and physical morphological data, you can overlap that data effectively to get not only the placement of the phylogenetic tree, but also the timings of the splits and branches. Nowadays, we also use what's called relaxed molecular clocks because in the past, it was thought more that molecular rates were fairly set, and now we know that they can shift. So we have equations that take into account the fact that mutation rates might be changing over time.
The equations with built-in wiggle room.
Exactly. So all of this is a tool for estimating what the most likely scenario is. This is why we will often say, based on molecular data, this species should have shown up around this time. The earliest fossil we have is here, but based on the genetic data, probably isn't when the species showed up because genetics suggest it was before that. And the real answer could be anywhere between those two based on we might have our mutation rate slightly off, that their rate shifted dramatically at some point, or the fossil we found is not close to being the oldest and there's actually one much older that then will adjust our info. But this gives us a ballpark to be aiming for and to expect when we are looking at the ancestry of groups and traits and things like that. We can also use things other than DNA, other proteins and other molecular features that would also be affected by mutation rates. So we can also look at the divergence of those structures. And with that, we can start wrapping up there's, mutation is one of those very foundational. So this is very much like a intro summing up.
Yes.
It is an extremely complex topic. So if there is a example of mutation or a topic related to mutation that we did not cover here, let us know, request the topic, and I'm sure we'll be able to get to it later on.
Yes, this is an episode that I think will pair very well with certain other episodes that we've done. So
Yes.
we did a genetics episode, episode 147. We did convergent evolution in episode 70. We did exaptation in episode
Yes.
78, which is a really nice go listen to episode 78 because we didn't bring it up very much in this discussion, but that really is like the next step in this discussion of mutation as it relates to evolution. You mentioned vestigial structures, which was episode 106. So there's a bunch of these that we can sort of cobble. Maybe we'll make like a genetics and evolution
Ooh, yeah.
playlist as we build up these topic discussions among our episodes.
Yes, no, that would be fun. So yes, let us know if there's something about mutations that you were thinking about the whole time that we did not get to. But for now, we will wrap up our main discussion and we will get to the end of our episode first with our last section, which is the patron question. Every episode, we like to answer one of the questions from our patrons, which you can submit if you are a patron of a certain level. What's our patron question today?
Today is a very straightforward question from Hannah, who asks if you could cure, in quotes, one common scientific misconception, what would it be?
I like this question. I thought it was very fitting for this discussion since mutant is a very often misunderstood topic and definition. Uh, we didn't go over it at the beginning because I wanted to save it for here. A mutant is just an organism that has had a mutation happen. So any organism that has a mutated cell in it technically is a mutant. Typically, that gets referred to more like bacteria because
Hmm.
a mutation has now mutated the entire cell of that individual bacteria. So that is truly a mutant. My arm cell that mutated is a mutant cell. I'm not really a mutant because that's not going to affect how I function as an individual.
Yes.
But that's
This
all
is,
mutant means scientifically.
yeah, scientifically, mutant is just, yeah, there was a mutation. Colloquially, the term has been applied to all variety of different things, but when we're speaking about it in terms of genetics and evolution, all it means is that it there is some genetic trait in this individual that is atypical for its population. Right? We talk about this.
Yes.
This comes up a lot if you look at studies on bacteria or like fruit flies, where it's like, yes,
Yep.
the mutant strain that has this genetic quirk that means that their wings develop differently or something.
Yes. And so this misconception that a mutant individual will be at times monstrously, and crazily different is a misconception. And the fact that, as we argued, all genetic diversity on the planet Earth today is the result of past mutations. Ergo, all organisms are mutants depending on what gene you're talking about.
Yes.
Like, the genes we have different from chimpanzees make us mutants from the ancestors that we shared with them. The genes that I have from my ancestors make me a mutant from them. Just depending on what genes you're looking at, all organisms have mutations in their body that at one point were another gene. So
Yes.
it is a lot less exciting in the scientific sense, but it is also less likely to lead you to thinking horrific things about how genetics work.
Yes. Well, and this also dovetails nicely with what seems to me to be one of the biggest misconceptions that forms a barrier toward understanding evolution, just as a
Hmm.
concept, which is this very common perception that evolution is a random process. And we
Yes.
got to discuss this a little bit in this episode that there is randomness to the variation that shows up in features and in genetics across a population, but how that variation affects the trajectory of that population or that species isn't random,
Yes.
right? In much the same way that if you collect a bunch of, right, if you just like scoop a bunch of rocks off the bottom of a river, what rocks you pick up and what their sizes are and whatever might get a truly random selection of rocks. But if then you pour those rocks onto a colander, which rocks fall through the holes is not going to be random. You are going to be able
Yes.
to look at the colander and go, all right, I can predict pretty well which rocks are going to fall through the holes. The colander is that natural selection is a filter. It is
Yes.
taking all of this variation and then seeing how that variation affects interacts with the environment and interacts with the different community interactions between the organisms, which of those traits is going to persist through all of that interaction. That is not random. And in fact, in many cases, it is very intuitive to understand how that is going to progress forward. There are random
Yes.
aspects to evolution, there are random aspects to the ingredients of evolution, but the actual path it takes is not it's not guided, it's not intentional, but it also, like the direction a river flows, is
Hmm.
not intentional. The river didn't decide what direction to flow, but if you look at a landscape, you can figure out where the river is going to flow.
Yeah, there are no rivers flowing up the mountain.
Yes, that you can understand the forces and the factors that are influencing that. And evolution is just a fairly intuitive outcome of a bunch of what can sometimes be different random effects.
Yeah. So yeah, that's a good one that I would cure if I had the magical ability to. And there's tons of others, but that list would be a very long list.
Sure. And this ties nicely into this discussion, so it's a nice
Yes.
little end cap on this one.
Well, because I just so often you'll see like some example of an animal with a a deformation or something, and so or a particularly big animal, and someone will be like, "Oh, do you think it's a mutant?" It's like, well, not the way you mean it.
Right.
If that is due to a mutation, it's not it's not in the way you mean it. Like gigantism is the cause of a mutation because it is the cause of a tumor growth that
Mm.
causes hormone release in a weird way. But that's not this person isn't a mutant in the way you are thinking that their mutant ability is being tall.
Yes.
Like that's not how it works. It's a good question.
Thank you for that question, Hannah. And thank you, of course, to everyone who submits questions and supports us on the Patreon.
Absolutely. If you're interested in joining us there, you can find links down in the description. You can also find a link to the website and our blog post on this episode that will have extra goodies, links and pictures. And I don't know how many pictures of mutation, of the genes and stuff, but whatever pictures David can find.
I'll just have pictures of my favorite X-Men.
Yes, there you go.
just along the whole page.
Remember, we got Dragon Con coming up soon, and we just wrapped up Snake and Croc month. So thank you all to your support, and check out any of the live streams or anything if you missed them.
Yes. DragonCon, hopefully we will have details soon to announce.
Yes.
Also, speaking of other things that are going on, podcast fans, Leave It to Us is quickly approaching the end of season 1. And if you are a patron supporting Leave It to Us, you can submit questions for the mailbag episode that will cap off the season. So consider doing that.
Thank you to all of our new patrons here on this podcast and that podcast, and those who requested this topic. And top level thanks to our tippy top tier patrons, Sarah May, Daniel the Bug Lover, Jeff Ellington, and Bugs all the way down. And with that, I have nothing more to say about you. This is now another fun one of I knew a lot of the information that I needed to say going into this because I've had biology classes and genetic classes. But then I learned even more about mutations, and now here's another episode where if you ask me something about mutations that wasn't on this episode, I probably won't have an answer for you because
This was it.
This went through most everything I know about mutations now.
You'll notice I brought up the lactose tolerance thing a couple times because it's my one go-to example. That and snake venom. I did it. I used up all of the tricks in my mutations bag.
I had that because I know there's been newses where we have mentioned like convergent mutations that have happened and like other examples that have gotten used in past episodes. And I couldn't remember any of those while doing notes for this. So that's why I found the one about deep sea fish.
Yes. So if you have a favorite example about mutations that you learned from us, let us know.
Yes.
Let us know what we taught you.
That we knew before this episode.
Yep.
And until then, we release episodes every fortnight. So that is that's our mutation rate of the episodes.
Like molecular clockwork.
Yes. You will be able to track back when we split. You will be able to track back.
This is how I can tell when like episode 300 is going to come out because we keep it nice and consistent.
Bye, everybody.
Bye.
Thanks for listening to the Common Descent podcast. You can follow us on Facebook, Twitter, YouTube, and check our WordPress blog for pictures and links after each episode. Huge thanks to our patrons whose support helps keep this podcast running and who get access to bonus goodies on Patreon. The song you're hearing is called On the Origin of Species by Protodome, which we found at ocremix.org. Thanks again for listening. We hope you'll join us next time.